Hey, what's happening, guys? Rouge again, and today we're talking about is cell division, aka mitosis. If somebody were to ask you what cell division is like, real quick, it might look something like this. But it's actually far more complex. So when we look at cell division, we have our cell in interphase, we see the chromosomes form, they line up in the middle, and they're pulled apart until we get two different cells, and the cycle happens again. Okay, so first of all, why do we have to know cell division? Why do scientists research it? Why do we learn it in school? Well, there's three big important reasons why we study cell division. The first one is it's the growth of new cells. So at the point of your existence and my existence, we started off as a single cell, and that cell grows and divides into 2, into 4, so on and so forth, until we consist of trillions and trillions of cells. So the first reason is the growth of new cells. Number 2, it's to replace old or damaged cells. So just like a car, if you take good care of your car, just by using it, you're still going to get damage from it. Even if you take good care of yourself, your cells are still going to get damaged to the point where they need to be replaced. So then they're going to replace, your body's going to replace your cells by cell division. The third reason why we study cell division is because it's actually a form of a disease. It's actually what cancer is. Cancer is uncontrolled cell growth. So if we look at these cells right here on the edge, these cells grew and divided, and then they got the message to stop growing and splitting. These cells got the message to stop growing and splitting. However, cancerous cells, they don't know when to stop. So what happens is they form a cluster of cells called a tumor. And that tumor could eventually steal nutrients from these nearby healthy cells and deprive them of nutrition, okay? So the three reasons why we study this: growth of new cells, replacing old or damaged cells, and cancer is a form of uncontrolled cell division. So let's refresh your memory on the cell cycle. We started off with interphase. In interphase, there's 3 subparts. G1, the cell grows. S phase, DNA is replicated, and lastly, G2, the cell is going to grow more. After the cell grows more, we're then ready for cell division, or mitosis. In mitosis, there's 4 subparts of that. We start off with prophase, metaphase, anaphase, and telophase. All 4 of those stages together, they help divide the nucleus and the DNA that's in there. And then lastly, we have cytokinesis. Cytokinesis is division of the cytoplasm or everything else that's in the cell. So here are the notes that I want you guys to know. After interphase, G1, S, and G2, the cell is ready to divide into 2 identical cells, okay? Then we move into mitosis. Mitosis is division of the nucleus in 4 distinct stages: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and sister chromatids pair up to form duplicated chromosomes, and the nucleus begins to break down. So I think of prophase, pair up. In prophase, the sister chromatids pair up to form duplicated chromosomes, okay? Next, we move on to metaphase. In metaphase, the nucleus is completely gone. It starts to break down in prophase. Metaphase, the nucleus is gone, and the chromosomes line up in the middle. So I think metaphase, middle. The centrosomes, okay, those little churros that we talked about earlier on in the year, the centrosomes send spindle fibers that attach to the centromeres. Remember, the centromere is the center of the chromosome. After metaphase, we go to anaphase. The cell begins to elongate. It starts to become more oblong or egg-shaped, okay? And the sister chromatids are pulled apart to opposite sides of the cell. Lastly, let's go back real quick. In anaphase, I think the A, I associate that with apart or away because those sister chromatids are pulled apart, okay? Next, we go to telophase. The chromosomes reach both ends of the cell and start to uncoil back into chromatin. 2 nuclei reform. So I think telophase, 2 nuclei. Now, if you take a look at these 2 right here, If you take a look at prophase and telophase, they are the exact opposite process. In prophase, the chromatin condenses to chromosomes. In telophase, the chromosomes go back to chromatin. In prophase, the nucleus breaks down. In telophase, the nuclei reform. Okay, so they're the exact opposite process. After we have the DNA or the chromatin reach opposite sides, then we go to cytokinesis. And cytokinesis is division of the cytoplasm. What ends up happening is we get a cleavage furrow. Think of that cleavage furrow as like a pinch. And it completely pinches that cell into two identical cells, okay? So this is the notes that I want you guys to take for the cell cycle continued. And let me show you what this looks like. So we start off with 46 chromosomes in each cell. That's how many a human has, 46 chromosomes. During S phase, that gets duplicated to 92 copies, but since they're attached, it's still 46 chromosomes. And then through cell division, we end up with, again, 46 chromosomes, but they're single copies, okay? They're single copies. So why do we form chromosomes in mitosis? It's to stay organized. If I were to try to separate that ball of yarn, it's going to be a mess. However, if you organize it, it's easy to separate that, and then each one will become its own cell, okay? That's why we form chromosomes, only for cell division. And here's what the cell cycle looks like. So this would be the cell in interphase. You can always tell when a cell's in interphase by looking at the nucleus. See how it's kind of fuzzy? The DNA is real fuzzy in there. You don't see any clear defined structures. That's because the DNA is in its chromatin state, okay? It's uncoiled and it's loosely packaged in there. Well, in interphase, you can see the cell, it grows, it gets bigger. The nucleus will end up getting darker after S phase because there's going to be more DNA in there. Okay? Then after S phase, it's going to grow even more in G2. And now it's ready for cell division. And notice that I have two centrosomes here. Okay? Those centrosomes are going to be used to split the chromosomes apart. So now we move to prophase. In prophase, look what happens. That nucleus starts to break down, and now I have clear defined bodies in there. I have clear defined chromosomes. And here, I'm starting to see more clear defined structures. Now, why does it still kind of look like a mess? Well, that's because there's 46 of those bad boys. There's 46 chromosomes in there, and they're overlapping each other, just like in here. Okay? But you see it, a clear structure as opposed to that fuzziness. Okay? Now, when we go to metaphase, that nucleus is completely gone. The chromosomes are lined up in the middle, and the spindle fibers from the centrosomes are attaching to the chromosomes, or the centromeres on there, okay? Remember, M stands for metaphase, and you can see that there's chromosomes starting to line up in the middle of that cell. After metaphase, we go to anaphase. That's where these spindle fibers actually start to pull the separate sister chromatids apart, okay? And the cell begins to elongate. It starts to become more oblong. And you can see right here, it's getting this funky shape because the cell is being pulled apart. And it's going to continue. This is still anaphase. And after anaphase, those sister chromatids reach opposite ends of the cell. Okay, so they're going to reach the opposite ends. And then it's going to uncoil back into chromatin. So I could see this is telophase because this is one cell. The DNA has started to decondense or uncoil back into chromatin, and the nucleus has started to reform. Now when we go to cytokinesis, check this out. You could see that the cell membrane is starting to pinch in the middle. And this right here, this divot, that's called the cleavage furrow. And what's going to happen is that's going to continue to pinch all the way straight down the center until we get two separate cells. Okay, so this right here is the cleavage furrow. And finally, the cell splits into two cells. and each one is an identical copy of the other. Okay, if you take a look at these images, I want you to think, which of these cells, A, B, C, D, E, or F, would be a cell in interphase? Well, that would be letter F. Why? Because the nucleus is real fuzzy and I can't see clear defined structures in there. Which cell would be prophase? Prophase would be letter E, because I'm starting to see those clear defined structures, those clear defined bodies. That chromatin is starting to condense and coil to form those chromosomes, okay? And we see that in this picture here. Next, which one would be metaphase? Metaphase would be letter A. You could see that in the cell, the chromosomes are starting to line up in the middle. Which one would be anaphase? Number A is away. That'd be letter C. You can see that the cell starting to become oblong as the chromosomes are being pulled apart. Okay. What about telophase? Now these two are very similar, so take your time. Telophase would be B. I'm starting to see two nuclei reform, and the chromosomes are starting to uncoil back into chromatin. Remember, telophase and interphase, they are the exact opposite process, okay? And lastly, cytokinesis would be letter D, and I see the cleavage furrow starting to pinch straight down the center until we end up with two separate cells. All right, folks, that is it for today, and I'll catch you all next time. Adios.